Hello everyone, I'm Marine VIALAT. I'm a PhD student at GReD Institute, and my work focuses on the role of cholesterol and its derivatives in prostatic tumorigenesis using a pathophysiological model, drosophila melanogaster. So first of all, the prostatic carcinogenesis can be described as a three-phases pathological process with first the initiation, when a punctual oncogenic hit affects a cell, preferentially from the epithelium. Then it is followed by the promotion, lasting several years, during which the cells will accumulate genetic alteration and proliferate anosically, leading to the invasion of the lumen. The last phase is the progression, with the formation of a carcinoma due to the rupture of the basal lamina, and then the cells will get the ability to invade other compartments and form metastases. In men, it has been described that androgens are involved in the support of the secretory function of epithelial cells, and this physiological role becomes pathological in late tumorigenesis by its supports by both the hormone and the receptor. However, the impact of androgens in early tumorigenesis remains unclear, and it is necessary to develop a model to study these phases. My working model is the accessory gland of drosophila. Localized in the genital tract, it represents a functional and structural equivalent of a prostatic acinus composed of a monolayer of epithelial cells surrounded by a basal lamina. In this model, by genetic approaches reproducing multifocal oncogenic hits, we can mimic the first steps of tumorigenesis, which mean that clonal cells expressing the hits will be surrounded by wild-type cells, and they will undergo a basal extrusion, leading to the formation of tumors outside the gland. Ecdysone is the major steroid hormone in drosophila, and it's supporting the reproductive function of the gland. This steroid is produced both by the prothoracic gland and the accessory gland from the cholesterol, and is able to interact with its specific receptor, ecdysone receptor. So the experimental strategy is based on the downregulation of ecdysone synthesis only in clonal cells by interference RNA. In this model, the tumoral induction leads to clonal cells inside the epithelium and tumors outside the gland. The downregulation of intratumoral ecdysone synthesis doesn't impact the presence of epithelial cells, but induces a new type of tumors with the invasion of the lumen by tumoral cells after an apical delamination. Some cells undergo a basal delamination without extrusion and localize between the epithelium and the basal lamina, and finally a few clonal cells are able to complete the basal extrusion. By quantifying these phenomena, we can notice a significant decrease of the number of tumors outside the gland, and in contrast, a significant increase in the percentage of gland with intratumoral tumors, corresponding to the ectopic cells. We conclude that ecdysone synthesis is involved in tumoral progression, and so the formation of tumors outside the gland. So to conclude for this short talk, our model recapitulates the early phases of tumorigenesis, from the initiation to the formation of tumors. Ecdysone intratumoral synthesis is involved in this phenomenon, and ecdysone deprivation promotes a new type of tumorigenesis, which reproduces what happened in men. Our aim is now to characterize the mechanisms involved in this phenomenon and to study their relevance for human pathology. So thanks for your attention, and if you want to talk or learn more about my project, you can ask your question or we can have a meeting. It will be a pleasure. Thank you.